So CRISPR-Cas systems are in real-time revolutionizing biology. They are revolutionizing the way that we do biomedical science. Their therapeutic potential will take a while to develop, but scientifically the potential has already been realized. And so scientifically what CRISPR allows us to do is study very directly and very rapidly how genetic changes, either a gene being deleted or a gene being turned on, directly affect whatever disease or cellular activity you're interested in studying. It's a total revolution. So when we think about the clinical application of gene editing technologies, there are many hurdles that we have to overcome. One hurdle is specificity. So you want to make sure that you're cutting the right DNA sequence. You don't want to cut the wrong DNA sequence for obvious reasons. The second thing that's really critical is delivery. So right now it is extremely hard to get this amazing CRISPR-Cas system to the right cell type. And so the clinical applications that we're likely to see early on are applications where you can take cells out of the body, edit them directly in a plastic dish, and put them back in. So these are diseases of like the immune system, for instance. You can take your blood out and isolate immune cells from your blood, edit them appropriately, and put them back in. But if you want to do gene editing in other tissues like your heart or your lungs or your liver, it's much more difficult because you have to target that CRISPR-Cas system to the right cell in the body, and that is extraordinarily difficult. So when we think about CRISPR-Cas9, it's clear that this technology is revolutionary, but it's really built on technology, other gene editing technologies that have been developed for a long time. So people have done fantastic work designing what are called meganucleases, which do gene editing, zinc finger nucleases, and also talons. At this point, it appears that CRISPR-Cas9 is easier to use than those technologies. Yeah, so for me, it was a really exciting opportunity to come to Georgia Tech. The bioengineering and biomedical engineering infrastructure here and the researchers here are world-class. It has to be one of the best departments in the world. And it felt like it was a place where I could share my technology with people and they would actually use it to do cool science. So at the end of the day, my lab is most interested in developing tools that other scientists can use to do biology. You know, we use chemical engineering and nanotechnology to hopefully make other people's biological research and medical research easier. That means that I have to have collaborators around me or I'm not being helpful to anyone. And Georgia Tech had a wonderful collaborative atmosphere, excellent students, and there are other people around the city at CDC, Emory, and so on that could potentially use some of our stuff.